Biology students, this is 10.2 part 2, and you want to continue watching.
Alright, let me bring up my 4x4 square. For our second example, we're going to do a dihybrid cross. This is similar to the first example in a monohybrid, except for in a dihybrid, we're going to use two traits. In this case, we're going to use color and shape of pea plants. For color, the dominant color is yellow, so we're going to use a capital Y. The recessive trait is green, so we're going to use a lowercase y. For shape, the dominant shape is round. We'll use a capital R. The recessive trait is wrinkled, so we'll use a lowercase r. In order to fill in these squares, the easiest thing to do is to look at the parent. In this example, we are going to use a heterozygous parent for both of them. Remember, heterozygous means different, so they have two different alleles, or two different letters, capital and a lowercase. To figure out what goes in the squares, the easiest thing to do is to see the combinations. Your first combination is a capital Y, capital R. Your second combination is a capital Y, lowercase r, lowercase y, capital R, and a lowercase y, lowercase r. We'll have the male for the top ones. For the female, she's going to have the same possibilities, since they're both heterozygous for both traits. Let's fill in this chart. First, let's fill in this first square. The first square, we are going to use this one and this one. Here, we have a capital Y and a lowercase y, so we're going to put both of these in the square. Yep. They are both capital.
So we can start right there, yes?
Yes.
Yeah. For this square, we are going to use the top one and the top left one. Here, we have two capital Y's and two capital R's. Following the pattern, these two will be used in all of the squares going down, and these two will be used in all the squares going across. Let's do all the squares going down first. Here, we have a capital Y and a capital Y, so we're going to write both of those in the box. We have a capital R and a lowercase r, so we're going to write both of those in the box. Following the same pattern, here, capital Y, lowercase y. Capital R, capital R, capital Y, lowercase y, capital R, lowercase r. Continue this pattern again, except for in this row, we're going to use these two. Capital Y, capital Y, capital R, lowercase r. Most commonly, the capitals are written first just to make it easier. Capital Y, lowercase r, capital Y, lowercase y.
And you can see the problem I have with her. So does this look like a capital Y? Yes. But she also said this was a lowercase y, and that kind of looks like that there. So if you're not really making it predominant, I can't tell, you can't tell, then I can't grade it, right? Okay, so make sure you do a better job than this lady who got paid lots of money to do this video. Just saying.
Capital R, lowercase r, capital Y, lowercase y, lowercase r, lowercase r. I'm going to just continue to write the rest of these for the following squares. From here, there's many ways to do it. This is the way I find the easiest. We need to figure out what these guys are showing as traits. We know anything with a capital Y is dominant, and those are going to be yellow. The only way to see a green plant is they both have to have two lowercase y's. So let's go through right now and write the colors. This right here, we know it's going to be yellow. I'm going to use a capital Y to show yellow. This one's going to be yellow since there's a capital Y. That one's yellow. So anything with a capital Y, I'm making yellow. These all contain lowercase y's, so these are going to be green, since I have two recessive traits and no dominant trait to mask over them. I use red. Let's look at the round and wrinkled. Round is being dominant, so anything with a capital R is going to be round. So doing the same thing, we're going to use a capital R for anything that is round. Anything with two lowercase r's is going to be recessive, and these are going to show wrinkled shape. Alright, now let's see our numbers. I'm going to erase this so I have some room. Alright, let's see what we got. Let's see yellow and round. So we're looking for anything with a Y and an R. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 of those. These are going to be yellow and round. Let's look at the ones that we have that are going to be yellow and wrinkled. So we're looking for anything with a large Y and a W. We have 1, 2, 3. Let's see what we have that is going to be green, which is a G, and round. We have 1, 2, 3. And finally, we're looking for something that's going to be green. Oops, hold on a second, that's supposed to be a G. Green and wrinkled. We only have one. It's located right here. Our ratio now is 9 to 3 to 3 to 1. This is our phenotype ratio. Phenotype being what is actually seen.
And at this point, I just want to remind you, if you haven't watched this video, it will be very apparent in class, and I can't help you. You will have to stay after for some sort of tutoring or come early for tutoring in order to watch the video before I can help you in class. That's why I show this ahead of time. So make sure you've watched it and you've told your friends to watch it and you understand the process so that when we get into class, we can dive deeper and I can answer your questions. When I get into these massive Punnett squares, like in a dihybrid cross, one of the ways I check my work is I go back and I add these up. 9 plus 3 plus 3 plus 1 equals 16. And as we can see, there's 4 squares on the left, 4 squares on the bottom. That's 16 squares. So that's just a real quick way you can do a self-check. So this is a stationary dihybrid cross for you to look at. 4 types of alleles from the male gamete and 4 types of alleles from the female gamete can be produced. And as you can see, it came up with the same ratio that we saw from our tutor. So probability. Probability is super important, and this is one of the places that people will often get have a hiccup. You can do the Punnett square correctly, you can do all that right, but you can really mess things up a little bit and make it muddy for the probability part. So the inheritance of genes can be compared to the probability of flipping a coin. Actual data might not perfectly match the predicted ratios. Mendel's results were not exactly a 9:3:3:1 ratio, but the larger the number of offspring involved, the more likely it will match the results predicted by Punnett squares. One of the things I do want to review with you, if we can come back to here for a minute, and we're talking about this. If I have a deck of cards, and I have 52 cards, how many queens do I have in that deck? Right, I have 4. Every time I shuffle it, those queens are going to be somewhere different. In one hand, I might find them all closer to the top. Sometimes I might get the first 2, and then I might get the 3rd, and then I'll have to wait till I get the very bottom of the deck to get the 4th. Through another shuffle of probability, I might get them all at the very end, and sometimes they may be evenly spaced out. There's no rhyme or reason, but we do know probability means out of those 52 times, we know that we're going to get 4 queens. So that's what you have to keep in mind. It's more of an abstract idea. So make sure you're good to go for test 9.2. Excuse me, 10.2.